Get ready to go. Hey. All right, the microphone is on. That helps it work so much better. I think I'm ready to go. Now that I got that problem solved, I'm happy to see so many of you came back, even after what I said all about glycolysis on Tuesday. Welcome back. Get ready for more. All right. Tuesday was all about converting a 6-carbon molecule, glucose, to 2 3-carbon molecules, pyruvate, and getting a little bit of ATP along the way. Net gain of 2 ATPs. You invest 2 ATPs, you get 4 back, total of 2 ATPs gained. For a lot of organisms, that's actually the end of the story in terms of getting energy out of glucose. 686 kcals per mole in a glucose molecule. A lot of organisms get just 15 or so kcals per mole back when they cash that in to make ATPs. Hideously inefficient, but 15 kcals per mole, 2 ATPs, beats the heck out of 0 ATPs if you need ATP, so it's a price that some organisms are willing to pay. There are organisms that are set up to do nothing but that. Again, hideously inefficient, but that's what's working for them because they can live in an environment where there is no oxygen. We are actually set up so that we can do that in times of need, during, for instance, anaerobic respiration, when you're weightlifting or something like that and expending energy faster than you can get oxygen to your tissues to finish off the job. But here's the thing. For today, I'm going to talk with you about the rest of the story, how it is you finish off that job, so that in the presence of oxygen, we can get as many ATPs out of each glucose molecule as you can, given the second law of thermodynamics, which insists that there's this entropy tax. So the deal for today is we're going to focus mostly on the citric acid cycle and the electron transport chain. Between the two of them, we finished the job of getting as many ATPs made from a glucose molecule as we can get away with. Before I get to that main point of the lecture, though, let me just quickly run through what's at the beginning here to remind you of the things we talked about, the highlights at least of what I talked about on Tuesday. These are redox reactions. Need I say more, right? Something's being oxidized, something's being reduced. Here what you're seeing is not so much a movement of electrons by tracking their charges. Rather, you're seeing movements of electrons from high energy states to low energy states. That means it meets the definition of a redox reaction. And the bottom line for what I was talking with you about on Tuesday is this chemical reaction. Again, we're going from glucose to pyruvate. Along the way, you're releasing a net gain of 2 ATPs, and you make 2 of these NADHs. It's only when I start talking with you about the electron transport chain that the wonder of NADH becomes apparent. So just be patient, it's coming. NADHs are valuable, not obviously yet though. Remember, this process, that arrow symbolizes a process. It's a process that's a 10-step process. There's an enzyme that catalyzes each step along the way. All 10 of those enzymes reside in the cytoplasm of eukaryotic cells. That's the compartment or the region of the cell where this business of glycolysis takes place. A little more detailed view. I want to just remind you, I'll say it one more time. Hopefully this will be it. Well, okay, one more time after this perhaps. You start the process of glycolysis by actually investing some energy. 2 ATPs are consumed to get the glucose molecule primed and ready to go through the rest of the process. A total of 4 ATPs get released or generated. They get generated through a process called substrate-level phosphorylation. Okay? And what that means in pretty simple terms is that an inorganic phosphate gets smushed into an ADP and that process creates an ATP. So it's substrate level phosphorylation. And for now, let me just tell you that that is in contrast to the only alternative, which is oxidative phosphorylation. Oxidative phosphorylation is what happens during the electron transport chain. That's where the NADHs really start to shine. We're getting to that. All right? So, but these ATPs get made by substrate level phosphorylation. And again, this is what's going on in the cytoplasm. But where we're moving today is to look more at what's happening inside the mitochondria. First, the transport of the pyruvate into the mitochondria in the first place. Then, how it is that the pyruvate gets converted ultimately to 3 carbon dioxide molecules. Each pyruvate is a 3-carbon sugar. Each of those pyruvates gives us 3 carbon dioxide molecules. By getting run through this citric acid cycle, ATP gets generated as part of that cycle within the mitochondria. But a bunch of NADHs get generated as part of this process within the mitochondria. And those NADHs get run through the electron transport chain, and oxidative phosphorylation takes place to give us a whole bunch of ATPs. This, but again, inside the mitochondria. This picture doesn't do it justice. It makes it look like there's roughly the same amount of ATP here and here as here. This needs to be about 10 times bigger than this or this, because that's where most of the ATPs get generated, at least for organisms that have oxygen to do oxidative phosphorylation with. Okay? One last look at the process of glycolysis as it plays out in the cytoplasm. Again, it's a 10-step process. I told you on Tuesday that I hope you'll be able to be comfortable with 3 different kinds of enzymes. Kinases. For kinases, they're transferring a phosphate group from one thing onto another thing. The first enzyme in the process is hexokinase. It takes a phosphate off of ATP and it tacks it on to glucose. Don't be worried about these names. Your textbook, I think, worries that you'd freak out by the names. It de-emphasizes them. For the citric acid cycle, the pictures that your textbook gives you doesn't even tell you the names of the enzymes. You shouldn't let those names intimidate you. They're pretty easy to figure out and to make sense of. The rule is pretty much this. If it's a kinase, kinase will be at the end of the name. And whatever it is that's the substrate is usually the first word. Sometimes it's the product. Usually it's the substrate. So here we have a kinase, because there's a phosphate group getting transferred. And here's the substrate. It's hexose, meaning 6. It's a 6-carbon sugar. Oh, we could have called it glucose kinase if you like, but for some reason we're stuck with hexose. They're roughly equivalent terms. That's what's going on with that name. So you got a kinase that adds some energy. You get an isomerase, another kind of enzyme I hope you don't get worried about. These are just changing the isomer, the structural formula. So an isomerase comes in next. You got another kinase that kicks into gear, right? That means a phosphate group's getting transferred here from ATP to fructose 6-phosphate to make fructose 1,6-bisphosphate. You get some more isomerases that are doing their thing, enzymes 4 and 5. You get the third kind of enzyme that kicks into gear for the 6th step in the process, the 6th enzyme, a dehydrogenase. When you see a dehydrogenase is involved, that means that a proton with its accompanying electron is either going to be added to or taken off NAD or NADH. Okay, so dehydrogenases always have NAD and NADH involved. Again, the substrate is usually the first part of the enzyme's name. What the enzyme's doing is the second part of the enzyme's name. That's the naming convention. Check it out. We got a kinase that's involved with the 7th step of glycolysis. The substrate is phosphoglycerol, right? 1,3-bisphosphoglycerate is over here. Actually, phosphoglycerol is the product. So this is one of those instances where the enzyme's name is the product followed by the kinase bit, as opposed to the substrate followed by the product, by the kinase bit. But again, as a kinase, you can see ATPs are getting changed. Got another kind of isomerase going on here, small modification taking place over there, and we end with one more kinase that creates the product pyruvate. So this is pyruvate kinase, and again, a phosphate gets transferred. All of those are substrate level phosphorylations. We get a net gain of 2 ATPs, just as we've been saying all along. So there you go. When you listen to the integrity recording of this lecture, you could skip perhaps all of Tuesday's lecture and just go with the first 6 minutes of today's lecture. That's Tuesday's lecture in a nutshell. At a fast pace, not for the uninitiated. Right? So you could hurt yourself if that's all you listened to. But I think in light of Tuesday, just gets us all right back where we were at the end of class on Tuesday. And this is the outline that I showed you on Tuesday. And guess what? Now we are ready to talk about the second half of the outline. This is the citric acid cycle, largely characterized and researched by a fellow in the 50s whose name is, or was, Hans Krebs. I told you on Tuesday, I'll stick by this story today, that Hans Krebs learned the lesson of the Davison and Danieli membrane guys, right? He was modest. He didn't want people to call this the Krebs cycle. He insisted people refer to this as the citric acid cycle. Despite his protestations, a lot of people prefer to refer to the citric acid cycle as the Krebs cycle, just in tribute to him. You'll hear it both ways. They're the same thing. The Krebs cycle, the citric acid cycle, all one and the same thing. In a nutshell, what happens with the citric acid cycle is we're converting something called acetyl-CoA to carbon dioxide, right? That's the bottom line. Along the way, some ATP will be generated through substrate level phosphorylation. But also along the way, a bunch, and you'll see exactly how many in a few minutes, but a bunch of NADH gets made, okay? And where the NADH really shines is what's going to come next when I start talking with you about the electron transport chain. But before we get there, let's talk about where the acetyl-CoA comes from, and let's talk about what's happening with this acetyl-CoA as it goes through the citric acid cycle. I'll steal my own thunder. There's not much to steal. This is kind of bookkeeping sort of stuff here, but remember, with glycolysis, we finished with this 3-carbon thing called pyruvate. Now, we're starting with a 2-carbon thing called acetyl-CoA. What's got to happen first is there's going to be a conversion of the pyruvate into acetyl-CoA. So, whoops. Here's what's going on. Remember, we ended with a 3-carbon thing called pyruvate. Now, we got to get pyruvate from that last slide, this thing, into the mitochondria. I've said it a few times. I think it's important I say it one more time right here. Glycolysis is in the cytoplasm, the citric acid cycle, the Krebs cycle, exclusively within the mitochondria of eukaryotes. Okay? The two shall never meet, right? Two completely separate processes taking place in different spots within eukaryotic cells. Mitochondria are specialized compartments that are set up structurally and with their chemical environments to facilitate this process of the citric acid cycle. The cytoplasm is the proper environment for glycolysis to take place. All right, so we got this slide that's showing you something that needs to happen. You need to get the pyruvate from outside the mitochondria to inside the mitochondria. There's a physical barrier, actually physical barriers. There's the double membrane of the mitochondria. It's not just one phospholipid bilayer. There's a double bag here for the mitochondria. that you'll probably recall. So there's a physical barrier. There's also a concentration barrier. You're going from low concentration, typically, of pyruvate in the cytoplasm to higher concentration inside the mitochondria. So there's a physical barrier. There's a concentration gradient you've got to run up against. This is not likely to be something that's going to happen of its own accord. Okay? In other words, the delta G for this, getting pyruvate from the cytoplasm into the mitochondria, the delta G is going to be positive. It's not a spontaneous reaction. Okay? How do you get non-spontaneous reactions to take place? Well, you link them to chemical reactions that are more energetically favorable than the unfavorable thing is unfavorable. Right? So you have to link this positive delta G thing to something that's got an even greater negative delta G in terms of its magnitude. And the bottom line is that gets accomplished by converting the pyruvate to a 2-carbon thing called acetate. If you look at the difference in the structure between pyruvate and acetate, focus here on the red carbons, right, and the grayish carbon, I suppose, not so much on the coenzyme A. I'll say a few words about that in a minute. But check it out. The red thing here in pyruvate is the same as the red thing here in acetate. That and that. This structure here is acetate. Acetyl is the sort of shorthand that's being used. But so that's the acetate. The acetate's right over here as well. But what's happened is that you've lopped off this carbon with its 2 oxygens to release carbon dioxide gas. If you're doing bookkeeping of high-energy covalent bonds, note that we've lost 1 high-energy covalent bond in this process. It's the breaking of this bond that pays the entropy tax, that allows you to drive this process and make it an energetically favorable process. So the price that cells are paying to get pyruvate into the mitochondria so that it can do the rest of the process of the citric acid cycle, the price that cells are paying is whatever energy is associated with this covalent bond. Notice that what's getting released isn't taking anything of significant value in terms of the energy stored in those covalent bonds. Carbon dioxide, for our purposes, is pretty much the definition of a molecule that has nothing but low-energy covalent bonds. The carbon to oxygen bond, the carbon to oxygen double bond, these are things, we're not leaving any energy laying on the table as a result of this. We're using some energy to drive the process of getting the pyruvate in, and we're also releasing something that has no intrinsic value to the cell in terms of the energy that it has associated with it. So we've got our first release of carbon dioxide gas. There's nothing more. We've wringed every last little bit of energy out of it that we can, and it goes away. Hopefully for a plant to make use of, to make more sugar at some point down the line, but we don't necessarily care where that carbon dioxide goes to. We're happy to have gotten the pyruvate across the double membrane of the mitochondria and into the mitochondria. A transport protein facilitates that process. Just for the record, and just so there's no mystery about this, that's a very specific, very dedicated transport protein. It doesn't do anything but move pyruvate from outside the mitochondria to inside the mitochondria. That's the only job for this protein. It's a membrane-bound protein. It's very specific. Shouldn't surprise you to hear that's what proteins, it's part of the nature of proteins to be very specific. This is such a protein. But that's not it. That's not the whole thing. We gotten the pyruvate inside the cell, but at the same time, there's an opportunity to harvest some energy from it. What do you suppose, what kind of enzyme do you suppose is involved with this process? And the 3 of these enzymes are all, you can see, 1, 2, and 3. The 3 of them tend to hang out together and work as a complex. You don't do one of these things without the other two things happening, pretty much guaranteed. Right? What do you suppose the name of the second enzyme is? Your textbook doesn't think you can handle it, right? Take that as a challenge. Your textbook, in its picture, doesn't tell you the name of the enzyme. I don't think in the book it tells you the name of the enzyme. They don't think you're tough enough. You guys can do this. What's the name of that enzyme? Let's start at the back end of the enzyme first, right? That's a dehydrogenase, right? No mystery there, no surprise. I haven't pulled any rabbits out of a hat. You should have seen that coming. This is a dehydrogenase. We know it's a dehydrogenase because NAD and NADH are involved. Simple, easy. These enzymes get their names. Second name, the family name is the kind of enzyme it is, a dehydrogenase. The first name of the enzyme is either the name of the substrate or the product. Typically the substrate. What's the substrate for this enzyme? Pyruvate. Hey, guess what the name of that enzyme is? Enzyme number 2 in that picture? It's pyruvate dehydrogenase. Simple, easy, not that scary. You guys can handle it. You're tougher than your textbook authors are giving you credit for. So there's a dehydrogenase. It's working on pyruvate. Pyruvate dehydrogenase does its thing. Another enzyme comes along and it works on that tweaked acetate and such, and it adds this other thing called, coenzyme A. The long and the short of coenzyme A is it's a big, bulky thing, okay? And it just gets tacked on to the end of that carbon molecule, such that at the end of the process, we've got acetate. Here we're going to call it acetyl, and we're going to acknowledge the presence of the big bulky thing, coenzyme A. It's more or less just a handle for the rest of the enzymes to be able to latch onto and pull along, right? We've got our acetate, we'll call it acetyl, and we're going to remember that the coenzyme A is there as well. Coming along for the ride, we've made acetyl-CoA. CoA is short for coenzyme A. Big bulky thing, connected by that thiol linkage, right? But the real star where we want to focus our attention, what you want to keep track of for bookkeeping purposes are these 2 red carbons. Where are they going to go? When are they going to become carbon dioxide, right? What's going to happen here? That's what the citric acid cycle is all about. I think it was in the second class when I was talking with you about subatomic particles. I mentioned that you can use radioactive isotopes of molecules. They have the same chemical activities, but they have unstable atomic nuclei. Radioactive, they can break down. We can see where they've gone. Hans Krebs was all over using radioactive isotopes. He used radioactive isotopes of carbon to figure out what was going on in the citric acid cycle. And here, these red carbons, it wouldn't be such a bad thing if you pictured that red as being sort of like glowing radioactive red, right? And as we go through the citric acid cycle, pay attention to where the glowing red carbons are compared to the not so glowing, not radioactive carbons that you're also going to see as part of this process. That's what Hans Krebs did. And that's how he figured out what was going on with the citric acid cycle. So here we are, right? We started with a 6-carbon molecule called glucose. We made it into 2 3-carbon molecules called pyruvate. And now we've converted both of those 3-carbon pyruvates into 2-carbon acetyl-CoAs. And we've kicked off 2 carbon dioxide gas molecules as part of the process, right? That's the tale of the tape so far. What are we going to do with the acetyl-CoA? Well, or the acetate in particular. Short answer, we're going to run it through the citric acid cycle. Here is a very high-level view of the citric acid cycle. Let's just get a feel for the lay of the land right now. In the citric acid cycle, what's going to happen is we're going to take that 2-carbon acetyl or acetate. We're going to kick off 2 1-carbon carbon dioxides, right? So we're going to convert all of those 6 carbons of the glucose ultimately into carbon dioxide gas, a total of 6. Along the way, we're also going to make 3 NADH molecules and 1 FADH2 molecule related to, but different from NADH, and we're going to have a crack at 1 more substrate level phosphorylation to make some ATP directly from the extraction of the energy from that acetate molecule. Okay? So there's a bunch of dehydrogenases that are going to be involved. There's a kinase that's going to be involved, and we're going to be kicking out some carbon dioxide along the way. This is showing you what happens for 1 acetate. Remember, you've actually got 2 acetates in play for every glucose that you started with. So for each glucose, you're going to crank through the citric acid cycle 2 full times, so as to be able to get all of that carbon into carbon dioxide gas. Now, it won't surprise you at this juncture, I'm sure, but I'm going to take you by the hand and we'll walk through each step in the citric acid cycle in just a little bit more detail than we had before. I think it's important and helpful to focus on the energy producing steps. You know, what's the strategy? How is it that this is getting accomplished? And so try to keep track. You know, is this an energy-rich molecule? Is the substrate energy-rich? Is the product energy-rich? What's their relative energy richness? Have we harvested energy at this step? Does that make sense? Does it all hang together? And I, too, like the challenge personally of not being scared by the names of the enzymes. And so I'm going to draw your attention to a couple of those along the way, just because I know you guys can handle it. But let's start at the beginning, all right? The first step in the citric acid cycle, straightforwardly enough, tip of the hat to Hans Krebs, pretty obvious way to start. You got something called the citric acid cycle. Let's make citric acid, okay? How do you make citric acid? You make citric acid by taking a 4-carbon thing called oxaloacetate. Kind of rolls off the tongue. You may not use oxaloacetate that often in casual conversations when you're in the line at Kroger, but you know, not that scary. Oxaloacetate. You take a 4-carbon thing called oxaloacetate and you hook it up with your incoming 2-carbon thing, acetyl-CoA. CoA is not needed anymore. It can get used, reused over and over and over again. So the coenzyme A with its sulfhydryl group gets kicked out. And what we're left with is we've added the 4-carbon oxaloacetate to the 2-carbon acetate, and we end up with a 6-carbon thing called citrate. A little bit of an aside to tell you about naming and chemical names for chemists. If you're talking about the salt of something, you put 8 on it. If you're talking about the acid form, you say acid. So citric acid, the salt of citric acid is citrate. If you would rather not talk about citrate, you could just as easily, everybody would understand, nobody would think any less of you if instead of saying citrate, you said citric acid here. Totally equivalent terminology. Same goes for oxaloacetate. If for whatever reason you didn't ever want to talk about 8s, you could talk instead about oxaloacetic acid. All right? That doesn't sound as cool to me as oxaloacetate, but you could, it's equivalent. Oxaloacetic acid is the same thing as acetate. It's just here we're talking about the salt as opposed to the acid form. Simple stuff. But look what we got here. We got a 6-carbon molecule. Count them if you like, right? We got a lot of high energy bonds here, a lot of carbon-to-carbon bonds. definition of high energy. Nonpolar covalent bond, right? We got a lot of carbon to hydrogen bonds, like these right here. Nonpolar covalent bond, high energy bond. This citric acid, this citrate, is a high energy molecule. When you look at this molecule, and in your mind match it up against glucose, what do you think? If I were to ask you which of those has more energy, mental picture of glucose, hopefully you can see, oh, there's probably more high energy bonds in glucose. We've lost some of the energy along the way to get to this point. That's okay, because we've made some ATPs, but they're in the same ballpark, right? This is an energy-rich molecule. How does citric acid stack up to oxaloacetate? This has more energy in it than this. How does citric acid stack up to acetate? Obviously, this has more energy than this. All right? And so what's going to happen is we're going to go around a circle. We're going to take this high energy thing, and it's going to end up becoming a lower energy thing, ultimately back to being oxaloacetate that we can juice up by adding another acetate to and start the whole process over again. So again, I just want you to be mindful to notice the flow of the energy here. You're going from energy-rich things to less energy-rich things, and then you give them a kick to get them back up to an energy-rich level again so you can start the process one more time. All right, so we get an enzyme that brings together acetate and oxaloacetate to make citrate. Look where the blue things are. The blue carbons are the ones that were the carboxylic acids attached to the oxaloacetate. All right, they're at the end of the molecule. Look where the red glowing radioactive carbons of the acetate are. They get tacked on over here. And as we go around, keep your eye on, you want to place a running bet? Here, let's make this more interesting. Get some skin in the game here. What do you think is going to happen? We're going to be releasing some carbon dioxide gas, right? What do you think is going to come off? Is the carbon dioxide gas that comes off, is it going to be red? Is it going to be blue, or is it going to be black? Let's find out. All right? That was actually Hans Krebs' first question. It's like, I'm going to feed these cells radioactive acetate, and I want to know what comes off first. I have carbon 13, I've got carbon 14, and I got carbon 12. Carbon 12 for him were the black ones, carbon 13 were the red ones, carbon 14 were the blue ones. What comes out first when I feed organisms radioactive carbon-13 acetate? Well, there's going to be an enzyme that converts the citric acid into isocitric acid, or isocitrate. This is an isomerase. Not a big deal. We're just we're rearranging the chairs on the deck of this molecule. Not a big deal. There's no difference in the energy associated with the covalent bonds of this molecule relative to this molecule. That's a minor rearrangement of the structure. Not much more to see there. Uh-oh, but something interesting happens in the next step. Right out of the gate, you can see NADH is involved. You want to shout it out with me? Dehydrogenase, right? There's got to be a dehydrogenase there. Your textbook doesn't even give you the name of that enzyme. They are so certain it would freak you out to know the name of this enzyme. I'm going to empower you. You'll know more than anybody else who uses that textbook that you guys have for this class. This enzyme's name, its family name, is dehydrogenase because NADH is involved. No shock or mystery there. The substrate that that enzyme works with is isocitrate. Guess what the name of the enzyme is? That's isocitrate dehydrogenase. That's the third enzyme in the citric acid cycle. The thing that converts isocitrate to alpha-ketoglutarate is isocitrate dehydrogenase. I'm not going to ask you that on a test, but I hope that if I asked you the sort of a name, if I told you this is the name of an enzyme, here's a substrate, here's the product, here's what happens along the way. What do you think? Which of the following is a reasonable name for the enzyme? If I gave you isocitrate, told you NADH was involved, and alpha-ketoglutarate, I hope you would choose isocitrate dehydrogenase as opposed to hexokinase or any of number of other names I could give you, right? So we got an enzyme, happens to be isocitrate dehydrogenase, but we've extracted some energy from isocitrate and made an NADH as we also kick off a carbon dioxide. Okay? So we've broken, at the very least, one high energy bond, one carbon-to-carbon bond, because we're letting this carbon dioxide go away. As was the case before with the enzyme that brought the pyruvate into the mitochondria, take note that the carbon dioxide that's leaving isn't taking any energy of consequence with it, right? That's trash. You've milked all the energy you're going to get out of that. Kick it off to the side of the road. And in exchange for letting that bond get broken, we've driven the synthesis of an NADH. And we'll use that to good effect in just a little bit. Look at this molecule compared to this molecule. Which of these has more energy? Which has a higher Gibbs free energy? Which has more enthalpy, energy stored in covalent bonds? This guy's a 6-carbon molecule. This guy's a 5-carbon molecule. Right out of the gate, this one's at a disadvantage relative to this one, right? All of this stuff makes sense from an energy sort of flow perspective. We've harvested something that can be used to make us ATPs, and we've gone in an energetically favorable direction in the process. Now, alpha-ketoglutarate, it's no slouch. Look at that molecule. It's got nothing to be ashamed of. Be proud if you're alpha-ketoglutarate. That's a respectable molecule. Got a lot of high-energy bonds there. A lot of carbon-to-carbon and carbon-to-hydrogen bonds, right? Still pretty energy-rich. Notice what the carbon was that got kicked out. It was actually one of the carbons from the oxaloacetate. It's the oxaloacetate's carbon, the blue guy, that was asked to leave the party at this stage. Both of the carbons that came from the acetate are still associated with the star of the show at this stage in the process. Okay? Just keeping track of where the carbons are. Well, we're moving through a cycle. What's going to happen next? I find myself hoping maybe this will be another energy harvesting stage, because that's really what this is about. Those are the happy moments in the cycle. We've captured some energy we can use to make some ATP. I feel good. That's exactly what happened. The next step in the process is where we're tapping into that energy-rich alpha-ketoglutarate. We're going to the bank, and we're getting some more ATPs out of it. We don't get the ATPs directly, but we do get the synthesis of an NADH. And I keep telling you, you just wait. We're going to be happy to have all these NADHs when we get to the electron transport chain. So we convert alpha-ketoglutarate to succinyl-CoA. That coenzyme A shows up one more time. Out of nowhere, it got tacked on at this stage. As that's being done, it's catalyzed by an enzyme. How does it happen? An enzyme does it, right? Cliche answer. That enzyme has a name, by the way. Of course, it's going to be a dehydrogenase because NADH is involved. The first name of the enzyme is alpha-ketoglutarate, the substrate. So alpha-ketoglutarate dehydrogenase converts alpha-ketoglutarate to succinyl-CoA. As that process takes place, you break a high-energy bond, a carbon-to-carbon bond, and you kick off another carbon dioxide gas molecule. Nothing left on the table there. That's pretty much the end of the energy scale continuum that we have. That carbon dioxide's pretty useless. Get rid of it. You break one high-energy bond to do that, but look what you get in exchange. You get an NADH. You'll be happy you did, right? That's nice. And we're back now to having a 4-carbon molecule, right? We started with a 4-carbon molecule. We added a 2-carbon molecule to get a 6-carbon molecule. We released one of the carbons to get a 5-carbon molecule. Well, we've released one more still. Check out which one. It's the other one of the carboxylic acids from the oxaloacetate. And we're back to having a 4-carbon molecule. And it happens to also have coenzyme A associated with it. No slouch, right? Succinate, this 4-carbon thing here, energy rich, lots of carbon to carbon and carbon to hydrogen bonds, but less energy than what we were seeing with alpha-ketoglutarate, and less still than isocitrate, but a whole lot more than what's in oxaloacetate, right? So what's going to happen the rest of the way? We're going to extract more energy from this to convert it to that in a stepwise process. I again find myself hoping maybe this next step's going to be an energy harvesting phase. Maybe we'll get some more stuff that's like ATP out of it. What do you think's going to happen? I'm a happy guy. Look at that. We got some energy in the conversion of succinate or succinyl-CoA to succinate. Here, we're making ATP directly. It's part of the conversion from succinyl-CoA to succinate, getting rid of that coenzyme A. As part of that conversion, we directly make ATP. That is an instance of substrate-level phosphorylation. Okay? What's actually happening is GDP, guanine diphosphate, gets made into guanine triphosphate, but the energy of guanine triphosphate and adenine triphosphate is equivalent, so you can convert. It's effectively a bookkeeping sort of thing. We can easily convert GTP into ATP. You've effectively made ATP. The enzyme happens to use GDP as its substrate as opposed to ADP, but again, it's an energetic equivalent. There's a phosphate group that's being transferred. Do I even need to say it? I better. There's probably 2 or 3 people who are not on the same page as I am yet. Maybe this is the chance to get with the program. There's a phosphate group that gets transferred. That means the name of this enzyme is a kinase. Okay? What do you suppose the name of this particular kinase is? Why don't we call it succinate kinase? Not a bad idea. Captures the name of the substrate and the product. Doesn't pay any attention to the coenzyme A. But succinate kinase comes around and gets us a net gain of an ATP. Simple looking molecule here. Simple looking molecule here. This one's got less energy than this one, primarily because we've lost the coenzyme A. Do you notice what else we've lost in going from succinyl-CoA to succinate? Here, structurally, it was still possible to distinguish those red carbons from the other 2 carbons that came from the original oxaloacetate. Here, with succinate, I can flip this around, right? This end of the molecule is no different than this end of the molecule. I lose track at this point of which carbons were the carbons that I got from acetate at the beginning, and which are the carbons I got from the oxaloacetate at the beginning, right? Because this end of the molecule is the same as this end of the molecule, I can no longer say this is the acetate end and this is the oxaloacetate end. I kind of lose track of my naming or my labeling convention, but notice that the carbons that get kicked out are not the carbons, it's carbon dioxide, they're not the carbons that came from the acetate, they're the carbons that came from oxaloacetate when this party started. All right, so there was a kinase that did something. Succinates our substrate du jour. What's going to happen here? We're going to convert the succinate to another thing called fumarate. Check it out. This has more high energy bonds than this does. A dehydrogenase called succinate dehydrogenase converts the succinate to fumarate or fumaric acid. You make an FADH2. FADH2 is related to, but a little different from NADH. You'll see how in a couple more minutes. But this is energy rich, and we can make ATPs with it. if we invoke the electron transport chain and do oxidative phosphorylation. So we're putting money in the bank at each step along the way so far, except for these first two. So we got fumarate now. What has to happen is we got to make the fumarate into oxaloacetate so we can start the process again. The rest is kind of boring. Going to need to rearrange things so that we can get back to oxaloacetate. But we have one last chance to get some energy out of it before we throw in the towel and we get back to oxaloacetate. There's an enzyme, a dehydrogenase called malate dehydrogenase that converts malic acid to oxaloacetic acid. When we're back to this step in the process, we are ready to start the whole thing over again. Now, I've done my level best to make that sound kind of interesting too. I've tried to add some drama, some misanthropomorphism to give you, human sentiments going on here. It's pretty much cut and dry, boring chemistry, but that's what the citric acid cycle is. And again, I think the things to keep track of as you're looking at the citric acid cycle, what's the objective? The objective is to take these high energy covalent bonds, specifically the ones that are part of the acetate molecule at the beginning, and convert those high energy covalent bonds into things that will give you ATP, so that you can use that ATP to drive chemical reactions that otherwise might not have been energetically favorable. That's the objective. That's really what has to happen here. And the rest is all just window dressing to accomplish that goal. It gets done in a stepwise fashion, because if you did it all at once, it would be equivalent to an explosion, right? Explosions don't do anybody any good. What we want is a nice gradual process so that at each step along the way we can extract some of that energy, capture as much of that energy in the acetate's high energy bonds as ATPs as we can. There are four dehydrogenases that work along the way. There's a kinase. Each of these steps is all about making ATP and getting us ready so that we can do the whole process over again. Just goes on over and over and over. Every time you bring me an acetate, I'm going to hook it up to an oxaloacetate, and we'll do this process one more time. For every glucose, 2 acetates show up at the party. We crank this wheel twice for every acetate. For every glucose, crank it once per acetate, twice per glucose. So you're getting a total, for each glucose, you're getting a total of 6 NADHs, 2 FADHs, and 2 ATPs. Each glucose gives us 6 NADHs, 2 FADHs, and 2 ATPs when it gets run through the citric acid cycle. Okay? Here is a zoomed-in view of all of those things. I am not going to torture you by saying the same things over again, but if you'd like, these PowerPoint presentations are available on the pilot page of the course if you want to get a closer view of these pictures and really start to count the high energy bonds and put them under your pillow and that sort of thing. I suggest you use these next couple of slides. It's just everything I've been talking about, just a little bit more of a zoomed-in view. Okay, there's a question.
For the exam, are we going to need to know what carbons are moving through each of them?
So the question is for an exam, would you have to know the carbons as they're getting kicked out and moved around? I'm not going to ask you to memorize these structures. Just like for glucose and glycolysis, there are going to be people that you meet later in your academic careers who are going to be mean and nasty, and they're going to insist that you memorize all that stuff. I'm not going to ask you to memorize it. I tell you what, I think it would be nice if you could, if I were to give you side-by-side alpha-ketoglutarate and isocitrate, and I asked you which of these has more energy associated with its covalent bonds, that you'd be able to do that. Now, you could do that by having memorized the structure of isocitrate and also memorizing that's got more energy than this, but I think you could do it more easily, excuse me, just by looking at these molecules and saying, oh, I'm going to count the number of covalent bonds that are high energy here, and there's more than that are over here. All right? So I won't ask you to memorize the structures. I think just as a matter of principle, it would be worth knowing that the carbon dioxides that get released in the citric acid cycle are not coming from the acetate themselves. They're coming from the oxaloacetate. Okay, just as a high-level principle, but I'll tell you that's pushing the edge of the sorts of things that I think would be helpful for you to take away from this class. That's the sort of thing that I might ask if I wanted to have a reason to distinguish between people who get an A and people who get a B. If you know that level of detail, you deserve an A. All right? So I don't think it's essential that you know that, but it wouldn't be bad if you knew that. And again, there are going to be others that make you memorize all these structures. I still have vivid recollection of using a pencil and paper and writing those structures out hundreds of times to make sure that I knew what those structures were. Later, in other classes, it's going to become important for you to know these structures. I don't think you need to know it for our purposes in this class or the next class in this series. I will show you at the end of the lecture today why it is people will insist that you memorize these structures at some point. And if you want to start memorizing them now, I'm not going to get in your way, and it wouldn't hurt you. It's just I don't think it's necessary. So let's keep moving around the circle. Zoomed in view, back to oxaloacetate. That's the Krebs cycle, the citric acid cycle. This is here pretty much just to telegraph to you there's a lot of NADH that gets made with each turn of the citric acid cycle. Okay? Up until now, I've just been telling you over and over again, you'll be happy the NADH is there. If you're rooting for cells and their ability to make ATP, making NADH is really good news. Let's talk about why it's good news. This is kind of a cool story. It's good news because of what happens with the electron transport chain. So I think it's time for me to start talking with you about what we do with this pile of NADH that's been made. The short answer is we're going to run it through the electron transport chain. Here is a picture of a cartoon, at least, of an electron transport chain. It wouldn't be a bad idea to think of an electron transport chain, this electron transport chain, and it is the epitome of electron transport chains. It is the electron transport chain. There are other electron transport chains. This is the electron transport chain. It would be fair to think of the electron transport chain as sort of a bucket brigade. Okay? Remember the old movies when you have to put out a fire before the days of fire trucks and fire hydrants? What they'd have to do is there'd be a guy by a river or a lake who would fill up a bucket of water, and he'd hand it off to somebody else, hopefully somebody downhill. It's easier that way. It doesn't have to be, but you hand off your bucket. That guy hands the bucket to somebody else, and you can go a mile of people handing off that same bucket of water so you could dump it on the campfire or whatever to put out the fire. That was an efficient way, the most efficient way at the time, of getting water from a water source to a fire you had to put out. You could have had each guy run to the lake and get a bucket of water and then dump it and then run back. It's easier if you just have the guy take the bucket of water and hand it as opposed to doing all that running. Okay? An electron transport chain is effectively a bucket brigade. The buckets here aren't carrying water. The buckets are carrying electrons. Specifically, they're carrying high energy electrons. And as they get passed along the bucket brigade, the electrons lose some energy. With each passage from one bucket to the next, you have a little bit lower energy than before. That's part of what makes this an energetically feasible and favorable process. The electrons associated with NADH are high energy electrons. Who cares what the scale is here? It turns out it's kcals per mole, right? Who cares? Check it out. On this axis, an electron way up here has more energy than an electron way down here. All right? And the whole point is that you start with high energy electrons, and with each passage of the electron from one of the buckets in the transport chain to the next, the electron loses a little bit of the energy. There's a flow to the electrons going from high energy to lower energy along the way. At the end of the bucket brigade, and each of these different things, these are cytochromes, cytochrome B here, cytochrome C. Cytochrome just means it's something that's got a pigment associated with it. That's how it got its name. But these are each different chemical compounds. Flavin mononucleotide here, coenzyme Q, cytochrome B. Don't worry about the names. The point here is that with each passage through this bucket brigade, the energy of the electron that's going down this path is getting reduced in a stepwise fashion. With each step, a little bit less energy. Ultimately, there's a very natural flow from high energy to low energy. At the end of the process, where we started with high energy electrons, we end up with low energy electrons, and those low energy electrons get foisted off onto something else, specifically oxygen, to make the lowest of the low energy molecules that we talk about, water. Okay? The ultimate recipient of these high energy electrons is oxygen, and they get used to make water. Water is the low energy waste product at the end of the whole process. The ultimate recipient of the high energy electron is oxygen. It gets used to make water. Right? You can't do this unless you got oxygen to foist those high energy electrons off onto at the end of the process. Okay? Kind of a big deal. You have to have oxygen there to receive the electron at the end of the process. If you take oxygen out of the equation, you know what you're left with? You're left with low-level energy electrons hanging out with cytochrome A3. If cytochrome A3's already got its hands full with a low-energy electron, it's not going to be able to accept another higher energy electron from cytochrome A just before it. If cytochrome A can't foist its electron off onto cytochrome A3, its hands are full, and it can't take a low-energy electron from cytochrome C. And if cytochrome C is backed up, you see what happens. You have to have the oxygen there at the end of the day to take those low-energy electrons. Okay? This is, by definition, it's tautological. It's the word. It's the meaning of the word. This is an aerobic process. It's not an anaerobic process. This is a process that is aerobic. All right? You must have oxygen present for the electron transport chain to work. It comes to a screeching halt. You get backed up if you don't have oxygen to take those electrons away at the end of the process. So none of this works in the absence of oxygen. Okay? But when oxygen's around, you can have this flow of electrons. And you may be wondering, why does that flow of electrons matter? What's the advantage to that? Sure, I understand you go from high energy to low energy. What's the deal? Why is this a good thing? It's a good thing because living things have negotiated a deal with nature over the years, billions of years, whereby it says, hey, look, Mother Nature, I understand. This is a problem for you. You don't like it when electrons have that much energy. You'd much prefer those electrons to have not much energy. I'm going to help you out. I'm a good guy. I'm going to come and I'm going to help you out. Here's the deal I'd like to make for you. I'm going to scratch your back. You're going to scratch mine. You know what I would like? I would like to have hydrogens on one side of a membrane as opposed to another side of the membrane. That would make me happy. I'm going to make you happy. What do you think? Can you make me a little bit happy at the same time that I'm making you happy? Seems fair, right? I'm going to let you get these high energy electrons to a low energy state, and all I'm asking in return is for, as these electrons are flowing downstream, to let some hydrogens go from one side of a membrane to another. That's all I ask. All right? That's what the electron transport chains are doing. And Here is a zoomed out version of that electron transport chain. Look what's going on. As those electrons are flowing, what happens is electrons, well, as the electrons are flowing downhill, at a couple steps along the way, 3 for NADH, 2 for FADH, because the electrons with the FADH start out further downstream. They're lower energy electrons. At 3 steps along the way for NADH, as the electrons are flowing, proteins are moving hydrogen ions from the inner sanctum of the mitochondria, the innermost space of the mitochondria, out toward the inner membrane space of the mitochondria. So remember, mitochondria have that double phospholipid bilayer. What we're doing is we're moving the protons, hydrogen atoms, from the innermost space to that space that's between the innermost space and the outside of the cell. With me? I think I'm making this sound more difficult than it actually is. But it's pumping protons from the very center of the mitochondria to that still membrane-bound space that's outside of that innermost membrane. And you look what's happening, right? That's all we've done. That's all we've done. We're helping nature feel good about getting these high-energy electrons to a lower energy state. Nature loves that. The deal that we've worked out with nature, because we're helping them in doing that, we're helping nature in doing that, is we're getting some protons moved from here to here. That's all we asked. And then we come back to nature and we say, hey, nature, I'm so happy that I was able to make you feel good with the electron thing, right? We have a good relationship. We get along. We can work together. I'm so happy that worked out for you. Feel good about that? I'm glad. The electrons were high energy, now they're low energy. Good for you. But I notice you've got another problem. And I'm thinking I might be able to help you with this problem too. I helped you with the last problem. Let's help again. Right? Let's work together again. I notice that there's something pretty nasty happening over here. On one side of this membrane, I got a whole bunch of hydrogens. And on the other side of the membrane, I don't have that many hydrogens. You don't like that, do you? That fact that there's a concentration gradient. You would rather that there be the same amount of hydrogen on both sides of the membrane. I can help you out. I'll help you out with that. How did that happen? But I'm going to help you fix it. Oh, and it's really upsetting to you, isn't it? Because not only is it that there's more hydrogens on one side of the membrane than the other, there's this nasty positive charge on one side of the membrane and a correspondingly nasty negative charge on the other side of the membrane. It's not just a hydrogen ion problem. It's a charge problem that's going on here. That must make you really uncomfortable. I want to be the guy that helps you with this problem. So what I'm going to do is this. I'm going to set up a protein that spans that membrane, right? And all this protein's going to do is let protons go from the high side to the low side, back to where they started in the first place. I'm just going to let them flow back. That's the kind of guy I am, nature. And I'm sure you're going to want to help me out with something for letting you scratch this itch that's got to be driving you crazy. I'm going to take care of that problem for you. You've got to do something for me, right? You're going to want to. You're going to insist on doing something for me. Really, you want to do something for me? All I'm asking is this. For each proton that flows back across that membrane through my protein that I'm putting in there for you, all I want is smush together one ADP and one inorganic phosphate and make me an ATP. Small price to pay, right? Who's to care, right? One ATP here when you got this horrible problem of the charge gradient, of the proton gradient. Why not let us do that? Let's set this up. Deal? All right, it's a deal. That's what's going on. You got the electron flow that is being used to drive the transport of protons from the innermost space to the inner membrane space. You set up this really uncomfortable environment for nature, and then you let the protons flow back across the membrane through a specific protein called ATP synthase. What a clever name for a protein, synthase. It's making ATP. And as the protons move through, for every proton that moves through, you get to make one shiny new ATP. For each glucose, you made 8 NADHs. Remember? 8-ish, 7-ish. 8. 2 from glycolysis, 6 from the citric acid cycle. For each of those 8, I get to drive 3 protons across that membrane, total of 24. And for each of the protons I put across the membrane, I get to make me an ATP. 24 ATPs from the NADHs that got made from glycolysis in the citric acid cycle. Pretty good deal. I get a couple more for the FADHs that I made. I get a couple more ATPs from substrate level phosphorylation. At the end of the whole process, I get a net gain of something like 34 ATPs. And we can quibble, and you know, sometimes it's 32, sometimes it's 36. You get about 34 ATPs for every glucose that you convert into carbon dioxide gas. Again, from an efficiency perspective, it sucks. It's about 35% efficiency for the energy transfer. But if you need ATP, it's a whole lot better to get 34, 36 ATPs for each glucose than nothing. So we live with it. And that's how we extract energy from glucose. This process here at the end is called chemiosmosis. It's just referring to the flow of the protons back into the inner membrane being used to drive the crank that makes the ATP. The whole business here, when you're talking about the electron transport chain, which is pumping the protons out, and the chemiosmosis that's letting the protons come back in, the whole business is called oxidative phosphorylation. So we got 2 kinds of phosphorylation. You got the substrate level phosphorylation where you're making the ATP directly, and you got the oxidative phosphorylation, which is oxygen dependent, which is electron transport chain dependent, which is making the bulk of the ATPs during aerobic respiration. I don't know. There's nothing more to say. That's a pretty good story, isn't it? I don't know. Did you like the deal-making stuff there? I didn't feel like you guys were engaged with me during that. But, you know, a lot of people would just put that in much duller terms than I've given to you. So I'm trying to help you guys out. All right? So there's one other thing about this slide that I want to share with you. I'm excited to have the chance to tell you about. This protein here, ATP synthase, by many measures, is one of the oldest proteins around. It's one of the first proteins that living things settled upon and got working really well. This is a protein that's old and pretty central to the way that life on this planet works. It's one of the first proteins, probably, that's got worked out, and it hasn't changed very much in billions of years. This is, you know, it's in the top 3 or 4 in terms of the oldest proteins that we still see working today. All right? That's kind of cool. And I got to tell you, the more you get to know about this protein, better. The better you get to know this protein, you know, get to meet it, shake its hand, look at it, stare at it, look at it from a couple of angles, the cooler this protein starts to look. So this cartoon isn't doing this protein justice. The next slide's going to show you a little more detailed view of this protein. And when you see this protein, maybe it's just me. But again, I'm telling you, this is an old protein. It's been around a long time. It probably hasn't changed much since it first got worked out. Biology, structure, and function are pretty related to each other. I'm building up to say to you, when I see this protein, in even the slightly more detailed that you get from the next slide, I can't help but find myself thinking, nice job, God, right? Nice piece of work here. Whatever came up with this is a nice piece of work. Check it out. That's a little closer view of that ATP synthase. If you were sitting down with a paper and pencil and you were charged with the job of coming up with a little machine that could allow the flow of protons to drive the synthesis of ATP, isn't that what you would have come up with? A little machine, right? With moving parts, there are gears, there's things that are spinning. Whoa! Structure, function, intimately related in biology. I'm not going to tell you how it is we got there. Evolution. But however we got there, that's a nice piece of work, right? Cool design for this little machine that is synthesizing ATPs, and it's been going on this way for a couple billion years. I mean, check it out, even this little thing poking down. Are you thinking that's got to be just some sort of artistic license? Look at the electron micrographs. People can take pictures of these things. There are actual movies with electron microscopes where you can see, the protons are too small to see moving through the process, but you can see this arm ratcheting, and with each ratchet, you know there's another ATP that's being made. I mean, it's just wonderful stuff. But let's get back to the whole purpose of this lecture, the whole talk here. Again, the objective, very simply, is if we've got an energy-rich molecule called glucose, and we want to get some ATPs, right? You got the $100 bill, you need the single dollar bill to get some work done inside of the cell. The way we go about doing that, whether you're a prokaryote or a eukaryote, is you use glycolysis. If you're a eukaryote, you get this really neat opportunity to use your mitochondria to milk every last bit of energy you can out of the process by driving through the citric acid cycle and using the electron transport chain with oxidative phosphorylation. And you end up with a net gain of 30-some ATPs from the whole process. Glycolysis happens in the cytoplasm. Citric acid cycle and the electron transport chain happen exclusively in the mitochondria, at least for eukaryotes, and that's what it's all about. If you don't have oxygen, you're kind of screwed. You can't get nearly as much energy out of this business as you can if you did have oxygen. Remember, the electron transport chain only works if you've got oxygen to foist those electrons off onto at the end of the process and to make some water along the way. If you don't have oxygen, the best you can do is just be happy with the 2 ATPs that you get from substrate level phosphorylation in glycolysis itself. The electron transport chain is off the table. Forget about all that stuff. You actually have a problem with the buildup of the NADH if you don't have oxygen to foist the electrons off onto. That problem gets solved in a fairly complicated way, but you get your 2 ATPs, and along the way, what ends up happening is you make one of 2 different kinds of waste molecules. Different living things have opted for one of 2 strategies to get rid of, you know, you can't make carbon dioxide and bleed it off into the atmosphere. You make something else instead. Some things make a waste product called ethanol. Some things make a waste product called lactate or lactic acid. You and I, when we do anaerobic respiration and you can't run the electron transport chain and do oxidative phosphorylation, we end up making waste piles, heaps of lactic acid. So after anaerobic exercise, strenuous weightlifting, when you're training really hard, what you're doing is you're telling your muscles, I don't care that you can't get me all the ATPs that you could have out of glucose. I need ATP right now. Give me whatever you can get out of the glucose right now. I'll pay a price for it later if I have to. The price you pay for it later is you convert glucose to ATP, and you make a pile of lactic acid. And a lot of physiologists feel the reason that you get muscle soreness, one of the reasons you get muscle soreness, is because those lactic acid molecules form crystals that poke your nerves and make you feel sore the next day. All right? So that's a direct product of anaerobic respiration in animals like you and I. For things like yeast that don't make the lactic acid, they make ethanol instead, we find, as humans, a use for their waste product. Beer, wine, that's what that's all about. This is fermentation. So you can either do alcohol fermentation or lactic acid fermentation. It's fermentation because what's going on is you're making an energy-rich end product that only gets made in the absence of oxygen. So when you're getting energy out, when you're making ATPs out of glucose, if you can't get into the mitochondria and do the oxidative phosphorylation, you end up instead making ethanol or lactic acid. And I told you a little while ago, 15 minutes ago now, that I was going to tell you why it is other people someday may make you memorize the structures of the compounds in glycolysis and in the citric acid cycle. This slide is what lets me tell you the reason some people will make you memorize these structures. Glycolysis and the citric acid cycle. Remember when I first told you about metabolism, I showed you that worm maze of all of those chemical reactions. Glycolysis and the citric acid cycle are central to metabolism. It's a testament to the fact that these were probably the very earliest chemical reactions that living things on this planet were catalyzing. They got worked out very early on. It's a testament to the fact that, and I'll harp on this in the last part of the course, that prokaryotes are all about energy efficiency. It's all about energy. It's the job number one. These are old metabolic pathways. Everything else gets tacked on to that existing infrastructure of glycolysis and the citric acid cycle. And so when a bacteria sees an amino acid, the first sort of thing that comes to mind is, how can I get energy out of this thing? And it gets energy out of that thing. For amino acids, they get shunted into, they get channeled into, funneled into, if you like, somewhere along the way. Each amino acid will either enter in as pyruvate, it'll get converted into pyruvate or acetyl-CoA, or be directly used in the citric acid cycle. If you get a fat, the fats get broken down and channeled into glycolysis, either as glyceraldehyde 3-phosphate or as acetyl-CoA. Carbohydrates all get converted to glucose or something related to glucose so it can get channeled through this process. And these arrows go in the other directions too. If you need to make a particular amino acid, what you do is you reach into your citric acid cycle and you pull something out of the loop and you pump it back in this direction. It's not energetically favorable. You're going to need ATPs to drive that process, but you guys now know where those ATPs come from. All right? This is at the core of all of metabolism. Oh, and I talked with you 2 lectures ago, a week ago today, I think, about inhibition. Check it out. This whole pathway, great example of substrate level, of non-competitive inhibition, and also stimulation. Doesn't it make sense that the first enzyme in the process gets stimulated to work better and harder if cells have low energy levels? You know a cell has a low energy level when there's a lot of AMP around. Doesn't it make sense that the first enzyme in the process doesn't work that much when you've got a lot of citric acid around or when you have a lot of ATP around? So this one enzyme kind of controls the whole thing, and it keeps track of how much energy is present in the cell. If you have a lot of citric acid and a lot of ATP, we don't need to run this process that much right now. If you have a lot of AMP, you get the opposite message, and that one enzyme responds accordingly. I'm done. Have a great weekend. See you guys on Tuesday. We'll talk about where that glucose comes from in the first